Okay, so I think we'll get started. So welcome everyone to the 7th edition of our Society of Chemical Industry webinar lectures. So my name is Oliver Ring. I'm a process development chemist at AstraZeneca and also a member of the Young Chemists panel, and I'm going to be your host for today's session. We've also got Nessa Carson from Syngenta and also Fred Hancock, retired from Johnson Matthey, on the line, who are going to be helping out with this afternoon's Q&A session. So for those of you who don't know, the Society of Chemical Industry aims to break down the barriers between industry and academia, and we do this by organising various events and conferences, including today's webinar session. So just a bit of housekeeping. For those of you who are new to the series, all delegates are automatically put into mute mode, so we can't hear you. If you have a question throughout either of our two talks for today's session, please do type that into the questions tab on your GoToWebinar control panel. And we'll aim to hopefully prioritise as many of these as possible, but please do bear with us. It would obviously also be great if you could write where you're dialing in from in the world, and also the institution that you're affiliated with, because it's great for us to hear where you're all calling in from. So just a brief thank you to our corporate partner and sponsor for this series. So AstraZeneca, it's been great having them on board to help get the ball rolling with this series. So a big thank you to AstraZeneca. And then briefly, I'm just going to introduce a few other SCI events that we've got going on at the moment. So the SCI National Undergraduate Online Poster Competition, the poster gallery has just gone live as of today. So we've got about 35 presentations being presented by final year MChems and BSc students across the UK. So it's great for you guys to support undergraduates. Based on the COVID-19 situation, all the students have submitted their posters, and we've got audio recordings from each of those. So I think one of us will post the link to this into the comments tab for you guys to look at. Also, in terms of future up and coming webinars, next week we've got a discovery chemistry talk from Lars Barfaker from Bayer. Then in 2 weeks' time, we've got a fantastic, really exciting electrochemistry talk from Professor Kevin Lamb at the University of Greenwich. So that's definitely one to look out for. It's going to be a great talk. Okay, so we've got two speakers today. Our first speaker is Matthew Burns from AstraZeneca. So Matthew started off his chemistry career with an undergraduate degree at the University of Bristol. He then stayed on to undertake a PhD with Professor Angerval in homologation of Boronic Esters. At this point, he was awarded a Marie Curie postdoctoral fellowship at the Colorado State University in the United States, and then also Columbia University with Professor Tomislav Rovis, in which he worked on the preparation and reactivity of artificial metalloenzymes. At this point, he then moved back to the United Kingdom to undertake a postdoc with Professor Matt Gaunter at the University of Cambridge. And then since 2018, he's been working at AstraZeneca in chemical development. So I think without further ado, I'm going to pass over to my good friend and colleague, Matt Burns, who's going to be talking about the route design and manufacture of AZD 5718. So I think without further ado, over to you, Matt.
All right, so yeah, I'm Matthew Burns. I'm working at AstraZeneca as a process chemistry up in Macclesfield, and I'm here today to talk to you about a project that's recently been in our department. It's on AZD 5718. So it's a small molecule drug that we've been working on. Okay, so AZD 5718. It's been developed to treat cardiovascular disease, and it does this by inhibiting the flat protein in the body. So the flat protein is a mediator on the biosynthetic pathway in the synthesis of fatty acids. It's these fatty acids that cause inflammation of the blood vessels and therefore cardiovascular disease. So by inhibiting this protein, you can reduce blood vessel inflammation. So the drug is currently in clinical trials at the moment. So there's a large patient body with cardiovascular disease, and this is a drug that they'd need to take every day of their life. Because of that, it's going to be a very high volume drug product if it does make it to market. So we'd be required to manufacture multiple tons of this drug every year. So if we look at the retrosynthesis of this molecule, you can break it down into three constituent parts. You've got this small nitrogen heterocycle, is what I'm going to talk about today. We've got a chiral acid fragment and this Boronic ester component as well, which my colleague Jean-Marc will discuss later on. So when the project came into the department, we had a short tactical objective, and that was to deliver 730 kilograms of this heterocycle. We'd use that to manufacture API to support clinical trials. We also had a secondary strategic goal, which was to establish the route to this heterocycle, and that be used for commercial manufacture. So the earlier you establish a route to a drug, the easier the regulatory documents will be to prepare as you do your work. So when the project came into the department, we had 18 months between initiating the project and actually having to deliver those 730 kilograms of material to the Macclesfield pilot plant for manufacture of the API. So API is the active pharmaceutical ingredient. Now, in Macclesfield, the guiding philosophy for our department is select. So inherently, most chemical reactions will have a risk or a danger associated with them, and any chemical process that we develop has fundamentally got to be safe. Environmental is another one. So again, in the pharmaceutical manufacturing industry, we are always going to generate waste of some sort, whether it's carbon dioxide or chemical byproducts or side products. So the main metric by which we measure ourselves here in Macclesfield is by the reduction of environmental impact that we can give to our processes. Another important thing is that any process that we design, any route to an API, has got to be legal. And by that I mean we've got to comply with local regulation wherever the drug is manufactured. For example, you can't use things like DCM in most places around the world. But in addition, you can't infringe on anyone else's patents. We obviously want to make all of our routes to drugs as cheap as possible so we can pass those savings on to patients. And the main way we do that is by increasing the yields of our reactions, decreasing reagent and solvent costs, but also selecting cheap, easily available commodity starting materials. So I said at the start that AZD5718 is a drug that patients may take every day, and they might take it for 20, 30, 40 years. So the impurity profile of this drug is so important because those impurities can build up in people's bodies over time. So we place a lot of emphasis on controlling the impurities in our compound, and we put a lot of effort into making sure that none of those impurities that are generated in our reactions will track through to the final API compound. And the last of our guiding principles is throughput. This is really to do with the manufacturability of our process. And ideally, you want to develop a reliable process that can be manufactured at multiple sites around the world. That obviously is going to make it cheaper to manufacture because you're not having to use specialist equipment that you might only find at one pilot plant or one plant in the world. But it's also going to secure supply chains. If patients are reliant on your drugs and they have to take them every day, you want a good, strong supply chain. So ideally, you want to be able to manufacture at a few sites around the world. Also with throughput, you want to make sure that you're sourcing reliable starting materials and that they're not reliant on, say, the cycle of crops, for example. So when you work in medicinal chemistry, you might be making a few milligrams of drugs, and that's all you need to obtain some biological activity. When you do this chemistry, you'll use the standard sort of synthesis, so multiple round bottom flasks, you can do some aqueous workups, then vac everything down on the bookie to get an oil, and then you might purify the oil by column chromatography. You don't have that luxury on manufacturing scale. You're restricted by the equipment you have in the pilot plants, and these will be a set number of vessels. And also, although it is possible to do column chromatography on scale, it's very expensive, and it's going to limit the amount of drug you can make per year. So normally when we work on scale, we would like to isolate all of our final compounds and intermediates as solids. And really what we want is to develop good crystallization processes or salt formation processes that can purge all of those impurities along the way and deliver a higher quality API. So it's really in the name. I'm a process chemist. I don't just look at a chemical reaction anymore. I do look at the whole process from start to finish and how you can really accommodate it on plant. So before we started the project, there was an established route to this small nitrogen heterocycle in AstraZeneca. It starts from this carboxylate purity here, and we do an esterification reaction followed by a transamidation of the ester intermediate. Now this amide here will get out in a good yield, and it's a nice isolatable solid. We then do a two-stage cyclization, so we form an alkyl chloride, and then we do a base-mediated cyclization to give us this bicycle compound here. The next step is a hydrogenation of the nitro group to the amine. Now, using high throughput screening, we did the previous teams did look at a lot of ways where you could hydrogenate off this benzyl group. Unfortunately, we couldn't find any. So what they relied on was using triflic acid to remove this benzyl group. Now, this route was used for previous manufacturing campaigns, and it could deliver 41 kilos of this bicycle However, going forward, we needed a route that could provide more material. There's also a few significant drawbacks of this route. So the first one is our starter material. It's relatively expensive. But also talking to suppliers of this starter material, we found out that some of their batches had failed, or at least they had failed to meet the specification that we required to make good quality API. So it's an expensive and somewhat unreliable starter material. In addition, there's some significant environmental issues with this route. So this cyclization reaction over here, to get good quality material out, we had to use dichloromethane. But the most significant one was the triflic acid. So it was 8 equivalents of triflic acid were needed for this benzyl D protection. Now, if this drug went to market and at peak year sales, we've predicted that we would require 500 tons of triflic acid a year just to make the API. So that's a huge amount of toxic waste. It's just not acceptable to put a process out there like that. So if our objectives are to deliver 730 kilos of this material, we really had to redesign the route from scratch. So we worked through a staged process in AstraZeneca where we do route design, where we look at alternative routes to the molecule. Once we've selected a route, we'll take that into process design, where we select reagents, process optimization, and then finally manufacture. So initially, I'm just going to talk to you about the route design work towards this molecule. So one of the easiest changes we can make was just changing the protecting group. So going from a benzyl protecting group to an alpha methyl benzyl protecting group meant we didn't have to use 8 equivalents of triflic acid for the D protection. We could just use 1 equivalent of MSA. It worked well, and we thought, well, it's okay, but it's not great. You're still relying on this same expensive, unreliable starting material. And in addition, with the established route, all of those nice crystalline intermediates were suddenly not such nice solids or oils due to the introduction of this alpha methyl benzyl group. So we looked at 2 other starter materials for our routes. One was this 4-nitropyrazole, and one was this 3-carboxylate pyrazole. Now, they're both relatively cheap commodity chemicals. I'm going to go into these retrosyntheses a bit more in the future slides. But we also looked at some other alternative routes, starting from some more slightly unusual materials. Now, we did get a lot of freedom in the route design stage. But I'm not going to talk about these routes mainly because they didn't work eventually. We weren't able to obtain the desired compound from these starter materials. So if you did start from our original nitropyrazole with this ester fragment here, and you alkylated it with an alkyl bromide, you get a 1 to 3 selectivity for this compound here. So you can see here we've alkylated on the correct nitrogen, and we could cyclize this towards our target molecule. But in fact, we actually get the wrong regioisomer. However, we found that if you remove this nitro group, this alkylation suddenly goes in the favor of the regioisomer you want. So you can take this relatively cheap starter material and alkylate it with this alkyl bromide. Then you take the crude reaction mixture and you do an HCl workup to debulk, and then a basic workup to initiate cyclization. And then you could obtain this compound here as a crystalline solid. So in one step, or in one process, you could access this bicycle from a relatively cheap starter material. We then went on to look at ways to functionalize this bicycle. The most obvious way to introduce an amine group into a molecule is through a 2-stage nitration reduction reaction. Now, we screened a lot of nitrating conditions, and the only ones that we can actually ever get to work on this compound involved sodium nitrate and trifluoroacetic anhydride. The combination of those reagents generates this rather frisky and very reactive nitrating reagent. Now here, the trifluoroacetic acid anhydride, also works as a solvent. Now this intermediate compound is a solid and did precipitate out of reaction mixture when diluted at the end of the reaction. So we could take this and do a simple hydrogenation to get down to our target molecule. So it's a nice, simple set of reactions and relatively cheap reagents. However, there's some significant safety implications with this nitration reaction. The first is because you're doing in neat solvent, there's quite a large, we've noticed there's a really large exotherm. We don't think that'll be safe to take to manufacture. The other one was that we observed a lot of off-gassing of brown gas. So although it's a simple process, safety considerations said that this would not be possible to manufacture with this route. Another route we looked at was through an MBS bromination to install this, to make this our bromide. And then through several rounds of high throughput screening, we're able to identify an Ullmann reaction that could convert this our bromide into nitro compound. So whereas this first bromination worked really well and gave a very clean intermediate, if this Ullmann reaction gave a large number of impurities, so for example, we saw the DES bromo compound, starter material, product, as well as a dimeric product where your product would act as a nucleophile and couple with your our bromide. And the last route we looked at to functionalize this unfunctionalized bicycle was using a Minsky style amination. So here we took this Hauser reagent, so N-hydroxysulfonic acid, and this iron catalyst which acts as a radical initiator, and in the reaction mixture you form radical cations, and these can electrophysically aminate your compound. It's a really nice direct way to introduce a nitrogen group. There were some drawbacks. One is the large excess of Hauser reagent we had to use. Although it's cheap, we did identify a few safety considerations with it, such as under the basic conditions, you'll form hydrazine. Whereas that's fine in a well-ventilated fume hood, doing it on a 100 kilogram scale is a lot more dangerous. In addition, we noticed a lot of unidentified impurities from the reaction. Although conversion was good, the overall yield was very low at 30%, and there'd be significant isolation issues of isolating a water-soluble compound from an aqueous mixture. Now, it's not impossible, and it could be overcome, but it is a significant challenge. So we looked at some alternative strategies to get to our compound. So another route we looked at, again, started from a similar strategy of taking your pyrazole and alkylating it with an electrophile. Now here we're using this alpha chloroacetonitrile. This is a much more reactive electrophile, so you get a much poorer regioselectivity this time. But if you take this intermediate compound here and nitrate it, again under the slightly frisky nitrating conditions, you get through to this compound here. From here we do a global reduction with Raney nickel and hydrogen, and we convert the nitro group to the amine and the nitrile group to a primary amine, which under reaction conditions spontaneously cyclizes to form a desired compound. So it's a nice proof of principle. It's 3 steps and all the reagents are relatively cheap and the starting materials commonly available. However, we've got this significant issue with this one-to-one regioselectivity in the alkylation step, as well as safety implications of this nitration conditions. But we really like this global reduction strategy, so we sort of explored that in a little more detail, but starting from alternative starting materials. So if you start from 4-nitropyrazole, you've got no issues with the regioselectivity in your alkylation step. And you can alkylate really well with the alpha chloroacetonitrile. We then looked at doing a metallation reaction, so using an organometallic base to deprotonate this proton here and react with ethyl chloroformate to give us this intermediate, which again, after global reduction, should give us our target molecule. However, you'll notice in this molecule, the most acidic protons are these ones here, and we didn't actually get any product out from this metallation reaction. So we looked at using an alternative electrophile, and we found that this benzyl-protected chloramine worked really well. So again, you could alkylate your 4-nitropyrazole to give you this compound here, and again, you've got no issues with regioselectivity. Then by treating this compound here with LDA, you selectively deprotonate the most acidic proton, and you can react that with ethyl chloroformate. That gives you this ester intermediate here. And then we do a global reduction. Again, we hydrogenate off the nitro group to the amine and remove the dibenzyl groups. We actually found that using a little bit of potassium carbonate really assisted this cyclization reaction. So by this point here, we'd assessed about 5 or 6 successful routes to our target molecule, and we go into a route selection process. Here it's really important because the route you're selecting is going to be used to determine the commercial manufacture of the drug. So we did end up selecting this route. Now the yields weren't amazing. There's a lot of things good going for it. The impurity profiles for each step were reasonable, and we thought we could work really well with the chemistry there. In addition, this compound here was a solid. Although this compound here was an oil, we knew it had this nitrogen functionality, which would help us make a salt. And this hydrogenation reaction here was relatively clean and was conducted in ethanol. And from ethanol, we knew we could easily form the HCl salt of our target molecule. So the route design work took us 6 months out of the 18 months we had available. And after this 6 months, we identified the real problems with the chemistry before we entered process design. So the main problem with this first stage 1 reaction was the isolation. We were doing an acid-base workup. This second reaction was probably the most, we knew that there'd be the most work required in it because it was a cryogenic reaction. It's done at minus 78 degrees. So whereas you can do reactions at minus 78 on a pilot plant scale, it's expensive and it's going to really limit the number of vessels that you can use around the world. So we really wanted a reaction that was non-cryogenic. We had a few impurities in this reaction which we didn't know how to purge, and the yield was relatively low. Also, with this final cyclization, there's a few small impurities there, and again, we didn't know how well we could purge them. The yield was quite low, and although we were confident in the isolation, we hadn't formally developed an isolation process. So after a further 2 more months of just process validation on a 1 gram scale, we contacted a CMO. So these are contract manufacturing organizations, and they would help us with the manufacture of the compound. And the reason why we do that was because our pilot plant here at Macclesfield was full of other projects at the time, and we were at this quite a large volume. So we effectively rent out pilot plant space from another company. They said, yeah, brilliant, come along board. But we also, at that point there, we had to commit to a route. So after doing this sequence on a 1 gram scale, we sent our processes out to a CMO and asked them to order $1.7 million worth of starter materials, knowing that we've got another 6 months before they start manufacture to really get the processes optimized and well defined. So I'll now talk you through the process design and the process optimization work that was done here at Macclesfield, and then the manufacture that was done at our CMO for each of these stages individually. So this alkylation step, or stage 1 as we call it in-house, was a relatively simple chemistry process. You'd obviously do your usual things of screening solvents and bases, but in the end we found that we could run it in a single alcoholic solvent of IPA and with a small amount of base. 2 equivalents of base, and that generates this compound here. Now the reaction also does generate a number of impurities if you look at them. So at the end of the reaction, we do a hot filtration, and that's to filter off the solid potassium carbonate, and we do it hot to prevent this compound here precipitating out of the IPA solution. So we do this first reaction in vessel 1, and we transfer to vessel 2. We remove some of the solvent down to 6 relative volumes of solvent and do a seeded crystallization. Now that allows us to obtain a crystalline solid compound, which we can isolate by filtration. And it also purges all of the impurities that we observed in the reaction. So the average yield out at our CMO was 96% for this stage. That's really good for a large-scale reaction. The LC purity, so detecting other organic impurities by HPLC, was 100%. So again, it's a very clean reaction. And the assay of the compound by NMR was 99.8 weight %. So got some really good metrics out of this reaction. So the largest batch size our CMO ran this on was 150 kilos of nitropyrrozole, and that delivered 430 kilos of product. In total, they ran 7 batches and were able to manufacture almost 2.5 tons of this compound. Now the way they had their plant configured was for a 2-vessel process. They did 1 vessel for the alkylation reaction and then transferred through a hot filtration to a 2nd vessel for crystallization. So the alloxycarbonylation process was a little bit more challenging, and we had to do a lot more work in determining reagents for this. So early in the route design, we established that this lithiated intermediate could be generated in LDA, and it was chemically stable at minus 40 degrees and below, but would decompose at higher temperatures. Now, a bit of work as well also showed us that this is much more chemically unstable in coordinating solvents such as THF or MTBE, or methyl THF. So we actually ended up using the less coordinating solvent TBME, as that really suppressed the reactivity of this compound here and made it a bit more stable for use. Now you could generate a batch of this lithiated intermediate and then dose in ethyl chloroformate to generate your product. There's a number of reactions that are generated from this, number of impurities that are generated from this reaction. So the first 2 I'd like to talk about are these 2 ring-opened impurities. So this compound here comes from the ring opening of our starter material, and this compound here comes from the ring opening of our product. So you can see through pKa analysis of the protons in our starter material and product that our desired proton that we want to deprotonate is by far the most acidic. But then you've got these 2 other protons here on these rings that is well within reach of your LDA base to be deprotonated and initiate this ring opening event. So a simple fix for this is just by changing your base to the less reactive lithium HMDS base. And when you use lithium HMDS in this reaction, these two impurities are greatly suppressed. So another impurity we saw was this carbon-linked dimer. Now, after a bit of investigation, what we found is that at temperatures greater than -40 degrees C, when you had this lithiated intermediate in the presence of starting material, it'll do a carbon-carbon bond-forming reaction to form this compound here. So this told us that if we want to do this reaction, or if we wanted to generate a batch of this lithiated species, we'd probably have to do it at -40 degrees C or lower. And the final impurity that we determined what it was and how it was made was this carbonyl-linked dimer here. So we found that if you generate a batch of the lithiated species and dose to it the product, you'll do this lithiated species will react with the ester to form this carbonyl-linked dimer. Now, on a manufacturing scale, this is going to cause a significant problem. So in your vessel, you've generated a batch of your lithiated species, maybe on a 100 kilogram scale, and you want to charge in ethyl chloroformate. Now, you might have to do that over 6 hours just to stop a large exotherm. So in that vessel, you'll be generating product over the course of 6 hours in the presence of your lithiated intermediate. So of course, you're going to go form this carbonyl-linked dimer. So one way you can get around doing this was to dose this lithiated species into a vessel of your ethyl chloroformate, but doing that on a manufacture scale where you've got to transfer a chemically unstable organolithium at -40 degrees from one vessel to the next is challenging and not suitable. So these results really told us that the process was viable. The process was good, but we couldn't use ethyl chloroformate as our electrophile. So we looked at some alternative electrophiles. So ethyl carbonate we can actually use as an electrophile, but also as a solvent. So the lithium HMDS doesn't react with it. So we could have this as a solvent in situ. And that meant as you add your lithium HMDS to your pyrazole starter material, you generate your lithiated species, which would immediately react with the electrophile. And because there's a large excess of this electrophile, it greatly swamped out reaction compared to the lithiated species reacting with the product. So we could obtain this product in about 95% area by HPLC with small amounts of this carbonyl-linked dimer, but still a significant amount, and we knew it might be difficult to purge if you're doing a salt formation. These two compounds both share basic nitrogens. Another electrophile we looked at was taking our batch of lithiated species and bubbling carbon dioxide through it. Now this gave one compound very cleanly. We didn't see any over-addition because obviously you can't add to a carboxylate anion. After neutralization of the reaction mixture, this compound precipitated out to form a nice white solid which should be isolated in a good yield. Now the only issue with this chemistry is, well, firstly, you're still going to generate a batch of lithiated species at -40 degrees C, and the second one was that this carboxylate, this acid intermediate decarboxylated under palladium catalyzed hydrogenation conditions. So the last electrophile we looked at and settled on was actually Boc anhydride. So the product of this reaction has a tertiary butyl ester now, so it's a bit more bulky, and that prevents attack of your lithiated species to form this carbonyl-linked dimer. But in addition, because Boc anhydride doesn't react with lithium HMDS, we found we could have the electrophile in situ as well, so it greatly streamlined the process. Also, that allowed us to get away with doing the reaction at much higher temperatures. As you're not generating a large batch of the lithiated species, you're only generating it for a small amount before it's trapped by your electrophile, you can get away with running it at non-cryogenic temperatures. So the process we developed, or the reaction we developed, involved taking our starter material, dissolving it in 10 relative volumes of TBME, and then adding our electrophile, our Boc anhydride. And we cooled the reaction mixture down to 0 degrees, and we charged the lithium HMDS in. And that gave us about 95% area of our product, and about 3% area by HPLC of our ring-opened impurity that derived from our product. Now we did a little bit of work on the understanding of this reaction. So we did in situ IR monitoring. So this is the IR spectrum of the reaction over time, and you can see as some peaks grow and some peaks shrink. We selected certain wavelengths that would represent product or starter material, and it plotted those against how they change against the time of the reaction. So we look at this blue trend here. This was assigned to the formation of product. So as we start to charge lithium HMDS to our reaction, we can see that the product increases. At this point here, we pause the rate of addition of lithium HMDS, and you can see the reaction immediately stops. This tells us that it's a really fast reaction, and it tells us that the kinetics of the reaction are related to the rate of addition of the lithium HMDS. And this is actually really good from the safety point of view as well. If you ever see a large exotherm occurring, all you have to do is pause the addition of lithium HMDS and you've stopped your reaction. It won't run away. Now we held the reaction as well, and you can see there's no extra formation of product. But then once we start to re-add the lithium HMDS, formation of our product continues. So we've now got to finish off the process, and we do that by doing initially a water wash. By charging in water to the reaction, we decompose this this impurity here. And you can see it's actually a vinyl nitro, vinyl nitrile, and enone all in one molecule. So it's quite a reactive molecule, and it's not surprising that it does decompose under aqueous conditions. And we can remove the byproducts of this impurity in the aqueous wash. So we do one aqueous wash, and then we do a solvent switch from TBME to isopropanol. And we do that because we can actually do a seeded cooled crystallization of our product out of an isopropanol mixture. So we've got two ways to purify the compound. One is through an aqueous wash, and one is through a crystallization. Now we're able to isolate the crystals by filtration. And again, this process was transferred out to our CMO. So the average yield that they got for the process was 88%. And again, because we've got these two really good purification methods, the HPLC purity of the compound was 100 area percent. And again, the assay was really strong. So it's 99.6 weight percent assay. Now, the largest batch size that was ran out our CMO, they're actually able to do in their commercial scale plant, and that was 320 kilos of the starting material. I think there's about 3,000 litres of solvent in there as well. Something, big, really big scales here. In total, they ran 14 batches and were able to generate over 2.5 tons of compound. Now, this is a one vessel process if you want to do it in one vessel, but our CMO found it a lot easier to do the reaction in one vessel and then transfer to a second vessel for workup. That's also really good for throughput because it means that whilst they're doing the workup in one vessel, they can get on and start doing the reaction back in the original vessel. So it really allows you to stagger your reaction. It makes it quite a streamlined process. So on to stage 3 now. This is our global hydrogenation reduction followed by cyclization. So after screening of catalyst and solvent, palladium hydroxide on carbon in ethanol was the best combination for our hydrogenation. This converts our nitro to our amine and debenzylates this nitrogen. So after this first hydrogenation reaction, we do a filtration into a second vessel to remove the solid supported palladium catalyst. Now, initially in our route design work, when we used the ethyl ester, potassium carbonate was a good base for cyclization. We've now got a tertiary butyl ester, so we need a much stronger base. We actually had to use 2.1 equivalents of tert butoxide to initiate the cyclization. After cyclization, we form the dipotassium salt of our compound, so we neutralize it with HCl to form the free base mixture. Now you can see at the start of this second vessel operation, we do a solvent switch from ethanol to an ethanol-methanol mixture, and that's for two reasons. The first is, at the end of this process, we want to remove the KCl byproduct by filtration. If you do this filtration in ethanol, it's really very slow. But if you do it in an ethanol-methanol mixture, it improves the rate of filtration by a factor of 10. And the second reason why you want to do this azeotropic distillation is to remove water from the vessel. So obviously your palladium on carbon catalyst is wet, and you're generating water from the hydrogenation of your nitro group. Now, any water in this cyclization step can go on and form this acid impurity here. It does that by opening up the lactam. So we do this ethanol to ethanol-methanol switch to really reduce the water content of the reaction. You can actually plot how the water content of the reaction affects this impurity over time. So you can see the water acts as a stoichiometric reagent. So the more water present in your reaction, the larger the amount of your acid impurity. So we've done our second vessel operation, which is the cyclization, and then we filter off the KCl, and then we take this free base mixture into our third vessel where we do the HCl salt formation. So we add our HCl and IPA to form the salt of our product. And this salt formation also helps to purge impurities. So we did see about 0.5 area percent of this acid impurity, which we can purge down to less than 0.1%. Now, in some cases, you can end up with some of this monobenzyl impurity from your hydrogenation. So this is where the hydrogenation has not gone to completion. And in the cyclization, this would cyclize to this compound here. But again, our salt formation was able to purge this impurity down. And even if you have we did spiking experiments, so even if you have large amounts of this compound, even at 6 area percent, you can purge it down to less than 0.1%. So again, this was transferred out to our CMO where they ran it, and it's really impressive for a 3-vessel operation where there's a lot going on. You've got a hydrogenation of a nitro and a dibenzyl group, followed by a cyclization, followed by a salt formation. You get 91% yield. The LC purity of the final compound was 99.7 area percent. Now the assay was a little bit lower, about 96%, and the remainder of the mass of this was KCl, which came through from the filtration. That's not a massive problem, and we knew that the following stage would tolerate small amounts of inorganic salts. Now the largest batch size that our CMO ran this on was 270 kilos of starting material, which generated about 110 kilos of product per batch. In total, they ran 10 batches, and they were able to manufacture over a ton of material. And this was a 3-vessel process. There's still some significant problems with this stage. So you're neutralizing a very strong base with a very strong acid, and this is quite an exothermic process. In addition, you've got this filtration of KCl. Now we knew it took a long time in ethanol, so we switched to this ethanol-methanol mixture to speed it up. But still, on a 270 kilo scale in our CMO's pilot plant, this filtration took 24 hours. So it's not impossible and it's very doable, but it still slows down your cycle time for drug manufacture. So if you go back to our original objectives, we're able to deliver over 1,000 kilos of product relative to our 730 kilos that were asked of us. And the reason we over-delivered more was just because we didn't realize how good a yields we were going to get. When we bought our starter materials, we thought we were going to get about 80% yield per stage as a reasonable estimate, but actually we were getting near a 90% per stage. And this project was complete within the 18 months, and the material was delivered to Macclesfield on time. This 4-nitropyrazole fulfills a lot of our supply chain concerns. It's a commodity chemical that is used in other areas outside the pharmaceutical industry, and it's relatively cheap as well. Now, we're really happy with stages 1 and 2 as they were high yields, reliable processes. Interestingly, we didn't do a lot of optimization on the processes, so we didn't look too much at charges of solvents or charges of reagents. We tend to save that work for more focused package of a DOE, design of experiment work, which we're currently doing at the moment. Now, stage 3 as well, we're really happy with it. There's a lot going on there, and we get some really high yields, but there's this non-ideal throughput concern with this slow KCl filtration. And again, there's a safety concern with this rather frisky neutralization reaction. So afterwards, we actually went back to look at this stage 3, and we thought rather than doing a base-mediated cyclization, can we do a Lewis acid-mediated cyclization? So we used the high throughput screening facility here at Macclesfield to screen a variety of Lewis acids. So you can see here, this is the terp-butoxides that our controlled terp-butoxide. So you can actually see that titanium isopropoxide and aluminium isopropoxide both act as good mediators for the cyclization. However, we chose not to really further look at these because we know that titanium isopropoxide has got a bad habit of forming titanium dioxide, which is a very insoluble compound and would be difficult to remove from our product. It might make it very white, but it will still lower the assay of our product. And aluminiums tend to have can form emulsions on scale. So the other 2 metals we looked at, or metal salts we looked at, were magnesium chloride and calcium chloride. They didn't give us higher conversions, but they did give relatively clean reactions. And we actually selected calcium chloride for further evaluation. So from a high throughput screen, it doesn't look like the best, but we knew it had the best potential. So just the final slide here is what we came up with. So you take your starting material and you do your hydrogenation. Again, you take your nitro group down to your amine and you remove your dibenzyl groups. That's done in your hydrogenation vessel, which after filtration gives you this free base compound here. Now again, we found that you had to do another ethanol to ethanol-methanol solvent switch. And that this time is because we want to increase the solubility of calcium chloride. So calcium chloride is just a lot more soluble in methanol than ethanol. Now we found we could use sub-stoichiometric amounts of calcium chloride, but we needed a stoichiometric amount of base to mop up any advantageous HCl generated. Now at reflux for 24 hours, this affected the cyclization. After the reaction, you add in your HCl and isopropanol, and that forms the HCl salt of your product. But at this stage here, all the other reagents of your reaction are still in solution. So the calcium chloride is still in solution and the Honig base is still in solution. So when you do your filtration, you're simply isolating your final HCl salt product, and all your other impurities are removed. So when we did this on a 10-gram scale in Macclesfield, we can get a good 83% yield and very high LC purity. And again, here you can see the assay is much more improved this time. So it's a much more streamlined process with slightly better results. The yield's not as good as the 91% we saw for our previous base mediator cyclization, but we're doing a lot of work on this at the moment, and we hope to actually implement this chemistry in future campaigns for the synthesis of our API. So I'd just like to finish with my acknowledgements. So I'm Matt. I worked on the route design work in stages 2 and 3. I was joined with Lai and Dave, who also did a lot of chemistry on this project. So Lai worked on route design, and she developed stage 1, and Dave Perkins did route design, also developed stage 2 and 3 with me. Alex, Angus, Alan, James have all worked on route design, and David's been taking a lot of the outsourcing work. We've got a really good analytical team. So Ahmed led the analytical team. Chris and Dominica worked closely with us in the lab to really help support us to develop the analytical methods to show that we can really control the processes. We've also got a large amount of specialist skills here at AstraZeneca, which made the project possible. So I'd just like to finish there, and thank you very much for everyone. If you've got any questions, feel free.
All right, thank you very much, Matt. That was a really, really great talk. All right, I think we've got time for one question right now, and then if you don't mind, we'll move on to the next talk and then have a Q&A for both speakers at the end. All right, well, I think I better ask the question that's come up a couple of times. So first of all, from Jason Tierney's Anima Biotech, and also from Matt Blackmore at AZ, they were both asking about the alkoxy carbonylation step, and specifically whether you'd thought about developing that in flow.
Yeah. Yes, we had. So as I said, the main problem with using ethyl chloroformate was that you can't do it in batch. So I think the week we got that result showing that it was a no-go in batch, we did go to our flow team here in AstraZeneca and presented them with the problem. That not only is that this would be right up their street. So actually one of the issues I didn't talk about was there's three times more carbon dioxide goes into manufacturing bulk anhydride than ethyl chloroformate. So if you can move away from bulk back to ethyl chloroformate, you're making a big saving from carbon dioxide here. So our flow team knew there's a good potential for the reaction, and they've taken it on, and they have developed a process, an oxycarbonylation process, and they're looking at taking it to manufacture in the future. I'm not going to go into too much because that's really their talk for another day, perhaps. But yeah, we've put this into flow.
Sounds fantastic. All right, well, perhaps I'll pass back to Ollie and we can move on to the next talk, but thank you very much once again.
Thank you.
So great talk, Matt. Thank you very much. So our second speaker for today is Jean-Marc Henry. So Jean-Marc started off his career in chemistry with an undergraduate degree in France at the European Chemistry, Polymer and Material Sciences University of Strasbourg. And he also spent a brief period at the Nanyang Technological University in Singapore. At this point, he moved to Manchester University in the UK with Professor Jason Micklefield and Michael Graney to undertake a PhD in halogenation enzyme chemistry. And at that point, he then moved into industry with a brief period at Redx Pharma as a medicinal chemist. And then in 2018, he joined AstraZeneca as a process chemist as a senior scientist. So again, another one of my colleagues, and I'm actually absolutely delighted to pass over to Jean-Marc.
Yeah, thanks very much for the introduction, Ollie. So as Ollie presented, I'm Jean-Marc. I've started working at AstraZeneca just shy of 2 years now. And the first project I worked on was the same project as Matt talked about, so AZD5718. So that's what I'm going to talk about today. And you might have guessed from the title that at least one DEA Boronic ester will be involved. So on this slide, this is just to remind you of the structure of the API, so active pharmaceutical ingredient, that is 5718. Matt already told you about the background of this molecule, so I'm not going to insist on that. I'm just going to move on to the next slide. where you will see a partial synthetic scheme for the synthesis of this API. So you can see on the top half the synthesis of the heterocyclic amine that Matt presented. So now we are going to focus on the bottom half of the scheme. And so in this bottom half, and in previous manufacturing campaign, we were able to identify some room for improvements in places. One major opportunity we could identify was after the Suzuki cross-coupling, which is stage 8 in this instance. The reaction itself worked extremely well, but the problems lied in the workup, where we had to rely on numerous washes and back extraction after the end of reaction. And also the biaryl compound was crystallized out in the aqueous layer, and that meant that the cake was really wet with water, which led to extremely long drying time on plant, and that increases the manufacturing cost of the product. And the reason why we had such an involved workup was because in the upstream chemistry, we were forming this organic impurity. So in the THP protection of 3-methyl-pyrazole, we form 2 regioisomers, the desired one here, and an undesired one, which is not reactive in the subsequent chemistry, but is still carried through. And the reason why it's carried through is that we don't have any isolation point on this branch of the synthetic scheme. Matt mentioned before that we don't rely on column chromatography, and that's true. This is typically very much a last resort. And seeing as these products here were all oily products, that would have been the only way to actually purify them. We envisaged, so you can see that we used the Pinnacle Boronic Ester as a reagent for the cross-coupling chemistry further down the line. We envisaged that we could maybe identify a different boronate that ideally would be a solid boronate that we could isolate in order to have an isolation point here, which would allow us to purge this impurity ahead of the Suzuki cross-coupling and then greatly simplify the workup. So that was the objective of this work package, so find a suitable alternative, a solid alternative for this Pinnacle boronate, and develop its synthesis and isolation. So we had different approaches that we considered. The first one was changing the protecting group, so moving away from THP and see if we could get a solid intermediate that way. To cut a long story short, all of the intermediate derivatives that we tried varying the protecting group were all oily products as well. Some of them were solid, but led to some unwanted deprotection ahead of the deprotection step at the end of the synthesis. And on the other side of that spectrum, third butyl protecting group just refused to cleave when we wanted it to. The other approach that we considered was modifying the moiety on the boronic ester side of the molecule by utilizing different diols and see if that could change the physical property of the product. Again, most of them led to oily compounds, except for the boronic acid itself or the lithium salt of the boronic acid, which were both solids. Unfortunately, they were very unstable and they degraded via protodeborionation under benchtop conditions within a few days. Fortunately, we also identified the diethanolamine boronic ester, or DEA Boronic Ester. I'm going to say that a lot. Which was a solid, free-flowing solid, which appeared to be stable under benchtop conditions. So this DEA boronate became our point of focus as a potential alternative to the Pinnacle boronate that we've used in the past manufacturing campaigns. Fortunately for us, we had a bit of experience at AstraZeneca using DEA boronates. So I'm just going to give you a very quick background on this kind of molecules. They've been initially used as a way to quickly hydrolyze Pinnacle boronates into the corresponding boronic acid going via the DEA boronate, which hydrolyzes really fast under mild acidic and aqueous solutions. And so that's quite straightforward. Later on, they have been used directly as Suzuki cross-coupling partners, which made a great deal to increase the value of this kind of intermediates, and that was then further built upon to increase the yield and conversions. And something that is very practical about these DEA boronates is that you can see the equilibrium here between a boronic ester and the diethanolamine molecule with the DEA boronate. Well, if you pick the right solvent in which the DEA boronate would not be very soluble, you can drive this equilibrium by precipitating the product. I hope you enjoyed this animation. And this allowed us at AstraZeneca to develop a standard process for the formation of the DEA boronates that we are interested in. Just to talk you through it quickly, we would typically carry out the borylation reaction in whichever solvent is best for this particular reaction and particular substrate. If required, we would then change the solvent via distillation, solvent swap, into THF, methyl THF, isopropyl acetate, or CPME. Why these solvents? Well, typically because DEA boronates are very insoluble in this kind of solvent, so you're setting up the right conditions for crystallization further down the line. And then by charging the diethanolamine into your intermediate solution and holding it for 15 minutes, you can then filter your desired DEA boronate. And this process was applied to a whole range of substrates, which you can see here. And I'm just going to bring your attention on these two, which have been selected for the commercial manufacture of two other APIs, which are part of our portfolio. And you can see here that they do have some advantages over the other boronates that were used previously. So in the case of Verinurad, we were forming a pinnacle boronic ester as well. And the isolation of this intermediate was made by changing the reaction solvent by solvent swap into heptane, which works well. And solvent swapping is a very practical tool for the process chemist because we can make the most of solubility differences of substrates in different solvents, and that's how we can isolate some of our intermediates. It does require a significant amount of solvent though, so it means that you increase the amount of waste that you generate, and that has a negative impact on what we call process mass intensity, which is just a measure of how much waste you create per kilograms of API that you generate. So if you can avoid it, it's always better. And in this case, by charging the diethanolamine, we can afford the DEA boronate, which as a plus was of a much higher purity than the pinnacle boronate after isolation. Another example is with the lanabecestat synthesis, where we used a boronic acid as an intermediate. And this particular boronic acid tended to form trimers, so with two others boronic acid moieties. And that meant that it was quite difficult to determine the purity of that boronic acid. And that leads to, well, obvious problems when it comes to charging the right amount of boronic acid into the reaction, and also some legal problems if you want to set this particular intermediate as a registered starting materials with the authority bodies. So again, by charging the diethanolamine onto the solution of boronic acid, we could isolate the corresponding DEA boronate, which came as just one species, easy to characterize. That was scaled up onto more than a 200 kilogram scale. And in the subsequent Suzuki cross-coupling, where you hydrolyze the DEA boronic acid ester, back to the boronic acid, you liberate the diethanolamine molecule again, which acted as a palladium scavenger, which is quite beneficial because either that means you manage to achieve a higher purity product after this, or it means that you can bypass other palladium scavenging techniques that you would have had to implement otherwise. So they do have their advantages. And so I told you before that this standard process was tried out on all of these substrates, and it usually works really well, except in the case of this full pyrazole substrate here. And you can see that the one we're interested in is down here. In the case of this substrate for the 5718 project, we used the Pinnacle Boronic Ester as starting material for this standard process, which we know was quite impure with all the impurities we were generating in the upstream chemistry. So these impurities could have had an impact on the solubility of the DEA boronate and therefore preventing the crystallization and not driving the equilibrium forward. But we were still interested in it, so we devised a plan to try to get to this molecule in a straightforward way. And so we envisaged that a one-pod lithiation, borylation, and transesterification directly to this DEA boronate could be a nice way of doing it. So it starts out fairly similar to how the Pinnacle boronate was synthesized with the lithiation. That's oriented to this carbon-hydrogen bond due to the oxygen that's present on the THP protecting group, forming this first intermediate, which is quenched with triisopropyl borate to form this complex, which should have a negative charge here. Then this complex can be quenched with an organic acid to yield this diisopropyl alcohol boronic ester. And from here, we thought this would be a nice setup to push this transformation forward, as it's quite entropically favored, as you would use one molecule of diethanolamine and liberate 2 molecules of IPA by doing so. So we tried out these conditions with the legacy reaction conditions from the Pinnacle Boronic Ester synthesis, and it worked to some extent. We could isolate some of the compound in 26% yield, and we could see by NMR there was a significant acetate signal with the product. So this obviously come from the acetic acid, which we used as an organic acid to quench the intermediate. Whether this was acetic acid itself or lithium acetate or an acetate salt of the product, we were not quite sure at this point. Fortunately, I was quite slow at discarding my mother liquors, which is always when the cool things happen. And more compounds crystallized out from these mother liquors. So I could isolate another 10% yield, and this one was 98% pure. And by comparing the XRPDs for both solid fractions, we could see they were identical, which made us realize that in the first instance, we managed to isolate DEA boronic ester alongside with a lithium acetate coprecipitate. Now, this lithium acetate coprecipitate will need to be addressed at some point, but considering the low yield that we managed to obtain at this point, we felt that it was more urgent to verify that we could have a high yielded process before tackling the isolation of the pure product. So we started by exploring the chemical space, looking at different solvents and different bases. Methyl THF and TBME led to the best isolated yield, and the reason why we favored methyl THF over TBME is because we can carry out the preceding THP protection in methyl THF. So looking further down the line, in an ideal world, we would like to telescope these two reactions together so we have a much more streamlined process. So using the same solvent was an obvious choice to us. LDA as base led to better results than with N-butyl lithium, and lithium HMDS in this case did not lead any conversion at all. You can see that the yield was very nicely increased to more than 90%, so we were quite pleased with this. We could scale this up to more than 10 grams, and again, the product precipitated right after the end of the reaction. Again, it was isolated with a lithium acetate coprecipitate. So we were in a much better place at this point. There was still a few problems we wanted to address. So you can see here that we are using 34 volumes of methyl THF as solvent. So that means that we would use 34 liters of solvent per kilogram of substrate, which is quite a dilute process that we would be looking at. Typically, we tend to favor more processes between 10 and 15 volumes of solvent. Again, we try to use reactions that are as concentrated as possible to limit the amount of waste that we generate. So 34 volumes was way too high in our minds. We were also looking at cryogenic conditions. So Matt has talked about this before. It's technically possible to do this on scale. but not every contract manufacturing organization that we do rely on to carry out our processes have the capability to do that on their plants, which means that this would remove some flexibility for us when the time comes to choose one of these organizations. Aside from that, it uses also a lot of energy to carry out cryogenic conditions on scale. So if we can move away from that, it's always better. So we had a look at, after a surprisingly successful -20 degrees trial reaction, we had a look at varying the temperature between -20 and 20 degrees and looking at lowering the amount of solvent down to 14 volumes. And we also had a look at how long the lithiation should be kept on. And we found that 0 degrees with 14 volumes of methyl THF and a lithiation time of 60 minutes led to the best results. You can see that the yield has taken a hit somewhat, but the advantages of using much less solvent and non-cryogenic conditions were compensating for the yield loss. Again, this was scaled up on 10 plus grams, and the product was isolated straight after the end of reaction, again, alongside with lithium acetate. So at this stage, we were pretty happy with where the conversion and isolated yield was, and we needed to address the problem of the lithium acetate coprecipitate. So we thought of different ways that we could implement in order to bypass this problem. The most obvious one would be to have an aqueous wash after the borylation in order to just wash out the lithium acetate in the aqueous layer. That would mean that the boronate intermediate that you form after the borylation would be hydrolyzed to the boronic acid. And in this case, we know that the boronic acid in question is unstable. So we were not a big fan of this idea, and we had a nudge that it was not going to work out. But this is quite a received method when doing this kind of lithiation borylation and transesterification. So this had to be tested out. And indeed, it led to very, very poor results, and barely any DEA boronate could be isolated after the transesterification, presumably because of the instability of the boronic acid intermediate in that case. Another method that we envisaged was to isolate the DEA boronate alongside with the lithium acetate coprecipitate, as usual, and slurry this mixture in an organic solvent so that the DEA boronate would be solubilized into that solvent, and the lithium acetate would be left behind and could be filtered off. This is an unusual process, but it does work, and this was actually the first method that allowed us to isolate pure DEA boronate. And because of that, it allowed us to trigger a whole range of activities, like looking at the solubility properties of this DEA boronate, making sure that it would behave just as well, even better than the Pinnacle boronic ester in the Suzuki cross-coupling, and also looking at how stable it was over a long period of time. Nevertheless, we are looking at a process here that would rely on 3 different filtrations. So first one to isolate the mixture of solids, then a second one to remove the lithium acetate, and a third one to recover your product after crystallization in the filtrate. And filtrations are always bottlenecks when it comes to process chemistry, and it takes a very long time to carry those out on plant. So suggesting a process that relies on 3 different filtrations is bound not to be the most efficient and not the most elegant. So we had the third option that was clearly the preferred option, and it was to avoid the formation of lithium salts or solid lithium salts with the product. And so for this, we figured that by identifying the correct carboxylic acid, we could quench the reaction medium after borylation, just like we do with the acetic acid. And this would, in an ideal world, lead to a soluble lithium carboxylate at this point. Then by charging the diethanolamine, we could then form the DEA boronate, which could be filtered right away and be pure with a single easy filtration step. So we screened various kind of acids with various side chains, so small side chains, branched, aromatic, or long linear chains. And they all led to soluble lithium carboxylate, except for pivallic acid, which seemed to be very much the exception to the rule. And by switching acetic acid with pivallic acid, we could isolate directly a DEA boronate that was 97% pure in the same yield than we observed before. So this was quite promising. We scaled that reaction up to 5 grams and led to the same results. And at this point, we had a second small round of optimization, which was essentially really looking at what the actual concentration of the LDA was, and titrating it every time that we would use it. And the concentration had gone down a bit over time as we were using it, and increasing the volume of LDA that we would need to reach the number of equivalent needed. We could increase the yield back to 85% yield, but the purity dropped to 63%. And we would see that the lithium pivalate in this case had precipitated alongside with the product. And this was repeated a couple of times. And we would see regular lithium pivalate coprecipitation as well. So we seem to have quite a variable and unreliable carboxylate coprecipitation on our hand, which we needed to control and understand so that it could be avoided in the future. One way we found that worked quite well was to carry out a hot filtration once the DEA boronate was formed. At 50 degrees, the lithium pivalate remained in solution quite reliably. And the DEA boronate isolated from these reactions was quite pure and had a purity of more than 99%. We looked closely at the impact of the concentration and solvent system of the LDA, and we had to look at concentrations between 0.7 and 1.8 molar in different solvent systems, so THF-hexane, or we also use methyl THF as well. We found the commercial solvent system THF-hexane 1 to 7 to actually lead to best results, and we could also identify a lower concentration limit for the LDA, in that if the LDA is more dilute than 0.8 molar, the lithium pivalate would remain insoluble even at higher temperature. And that's because the lower the concentration of the LDA is, the more volume you have to add, which means the more hexane you add into your reaction mixture, and that decreases the solubility of lithium pivalate. Another solution that we found to be suitable was to increase the charge of pivallic acid. That leads to an increase in solubility of lithium pivalate, and the lithium pivalate would then stay in solution even at room temperature. And again, the DEA boronate isolated from these mixtures would reach more than 99% purity. The drawback of increasing the charge of pivallic acid or hot filtration is that the solubility of your DEA boronate is actually increased. So the yield, the isolated yield that you obtain from this goes from 85% back to 75%, unfortunately. Nevertheless, 75% is quite good, so we were quite pleased with this. And we aimed then at integrating that lithiation borylation with the preceding step, which is the THP protection. And this works quite well. So each step is roughly 75% yielding. So overall, that's 56% yield. This was then further scaled up onto 50 grams, and it worked just as well. That was, I think, the biggest scale that had been carried out when I created those slides. We're now in the process of further increasing the scale, and we'll be looking at more than 100 kilogram scale in a few months' time. So it's quite exciting to me, and I'm really looking forward to seeing the results from that. On this slide, we're just going to summarize the changes that were brought to this process using this very biased color scheme. You can see that we started with a Pinnacle Boronic Ester, which was an oil that was carrying quite a lot of impurity. It was generated in a very dilute reaction mixture at cryogenic temperature. And we have now moved away from this to favor a DEA boronate, which is a solid that is typically isolated with more than 99% purity. This is a typo. The reaction mixture is much more concentrated, and we are not relying on cryogenic conditions anymore. The only downside is that the yield is somewhat impacted, but we feel that there's enough advantages for that not to be a problem. Now, Matt introduced you to the concept of SELECT, which is the anagram for all these factors that we look at throughout the process development work, and we try to, that's what we keep in mind when we try to optimize a reaction. And in this case, if we have a look at the impact that this work had, we were able to switch to a solvent that comes from renewable feedstock, and we use a lot less solvent and also non-cryogenic conditions, so which means that it uses a lot less energy, so that has some nice beneficial impact on the environment footprint. The control strategy for the whole API synthesis is greatly improved because we have introduced a new isolation point, which allows us to flush out any impurities that is formed in the upstream chemistry. The throughput of the reaction is also increased because we can carry this out under much more concentrated conditions and under non-cryogenic reactions as well, so we don't have to spend a lot of time cooling down a massive vessel to very, very low temperatures. On this slide, we're just going to have a look at the impact that this have had on the subsequent Suzuki coupling. You can see that the conditions didn't change too much from the previous process to the new process. We just switched to an organic base. We are using the same polyimide catalyst, but we have been able to reduce the catalyst loading by 10-fold, which is quite significant. The solvent is now just water, so we are using less organic solvent, and the temperature and yield remain roughly the same. But if we look at the workup that comes after this Suzuki reaction, you can see that that's where the main differences come in. We used to be relying on 5 extractions with organic solvents in order to remove all the impurities from the upstream chemistry. That was followed by a crystallization in water, and the cake that was obtained from this crystallization required 3 cake washes to achieve the specifications we were looking for. And now with the new process, so by using a DEA boronate instead of the Pinnacle Boronic Ester, we have been able to reduce the amount of extraction from 5 to 2. The crystallization is now performed in the organic layer, and that allows us to reach the desired specification with only 1 cake wash at the end. Looking at the sum of the select factors that we talked about, you can see that a lot less solvent, especially due to the simplification of the workup, is used. So that means a lot less waste for the process as a whole, which is quite beneficial from an environmental point of view. The control strategy is also improved as the biorile that we isolate in the new process has a higher purity than that was obtained before. The throughput, again, is positively impacted because we spend a lot less time during the manufacture to perform the workup, and that also translates in a positive impact on the cost of the manufacture. The cost of manufacture is, of course, mainly reduced because we were able to reduce the catalyst loading by 10 times. So that's quite a significant improvement. So just to give you a bit of metrics, this DEA boronate Suzuki development work allows us to reduce the cost of goods for the API synthesis as a whole by 20%. And if we look at this work package and combine it with Matt's work package, so the development of the heterocyclic amine that he talked about, the cost of goods has been reduced by about 55%, which is incredibly significant and very important to ensure that this medicine, or any other medicines for that matter, can be made available and accessible to everyone that might need it. Another metric I can give you is the process mass intensity, so the measure of how much waste is generated per kilograms of API that you generate. With these 2 work packages, so this one and the heterocyclic amine that was presented before, we could halve roughly halve the PMI, so it went from roughly 560 down to 300. So again, a very nice improvement on that. And just to wrap things up on the DEA Boronic Ester, so the DEA boronates are a class of molecules that are usually very easy to generate and isolate from the corresponding pinnacle boronate or boronic acid, and they're usually free-flowing solid, which is why we do like them a lot in process chemistry. The standard process that we had developed at AstraZeneca had some limitations, mainly around pyrazole substrates, and so we addressed these limitations with a new process based on the soluble lithium salt, and this new process was selected for the commercial manufacture of 5718, and also because it enabled significant improvement in the workup of the subsequent Suzuki reaction. So I'd just like to thank everyone that was involved in this work, so all the chemists part of the 5718 team, and of course all the specialists that support us in this work, so crystallization scientists, analytical support, process engineers, and other kind of specialists like computational scientists, solubility, high throughput screening, and so on. So thanks everyone for your work, and thanks everyone for listening. So if you've got any questions, we can take them.
Once again, thank you very much for a great talk.
So shall I jump in first with the question, or
go for it?
Yeah. So we've got a talk from, sorry, question from. Apologies if I get any of these names wrong. Jahin Kim, no affiliation. Did you see any proto-deboronated adduct during the Suzuki coupling when using the DEA boron compound?
So there was a high throughput screening that was made for that, and we were looking closely at how much protodeborylation we might observe. That's a reason why we switched the base. So we went from potassium carbonate to DPA because it led to much less protodeboronation. We also know that if the Suzuki cross-coupling reaction was not ongoing, the product would protodeboronate, but the rate of the Suzuki cross-coupling reaction in this case is very fast, so it's complete within 30 to 45 minutes. So basically protodeboronation doesn't really have the time to happen in that case.
So I noticed on your slide, it said that you just did the Suzuki in water, is that correct?
Yeah, so that was not my own work. This is information I pulled from other people, but yeah, just water.
Cool. What scale was that on?
Good question. I'm not sure, Ollie, I'm sorry.
No, that's all right. Nessa, do you want to?
Yes, we've got a question coming in from David Stewart at Liverpool Chirochem. David's asking, is it possible to synthesize DEA boronates directly via a Miura borylation? Not sure I've seen that.
Oh, well, the short answer is I don't know, but it would be worth looking into because it would be a nice, straightforward way to get to them.
I think it would be really cool, but I don't know if the amine would just completely interfere with all the catalysis. Yeah. Yes, we got a talk from Daniel Blair. Sorry, a question from Daniel Blair. Did you ever consider using midoboronates? They've shown to be tolerant in aqueous workups and have good solvent anti-solvent behaviors.
Yeah. So the reason why we did consider midoboronates actually, but the reason why we never did much work on this for this particular project is because we were taking advantage of a carboxylic acid function to isolate the pyrite product from the Suzuki, and in the Suzuki with the midoboronate you would release the methylamino diacetic acid, which we thought might make it tricky. It might be tricky to purge this diacid because it should follow the organic acid that we were interested in. The other reason is that the conditions to form midoboronates are much more harsh than what we use to form the DEA boronate. So looking at the literature, we're looking at solvents like DMSO and DMF under DIN-star conditions, refluxing for more than 10 hours. And then the isolation of those MIDA boronates, if you're looking at doing it without column chromatography, is quite challenging as well. This is why we favored the DEA boronate.
Yeah, we've got a couple more questions about the boronates from Paul Blaney at C4X Discovery. He's asking he's asking about a comparison of the DEA boronates with both just ordinary Pinnacle boronates and trifluoroborate salts. And sort of what are the limitations of DEA boronates compared to them, and do you think that the others would have worked too?
So the comparison between the DEA boronate, Pinnacle boronate, and the trifluoroborate.
That's the one.
So in this particular case, between the Pinnacle boronate and the DEA boronate, in terms of performance in the Suzuki coupling, they were quite similar. I think the Suzuki coupling with the DEA boronate was faster because it would hydrolyze to the boronic acid, which is just faster to transmetallate during the polyimide catalytic cycle. With trifluoroborate, this is something that I've stayed away from as well. During the Suzuki cross coupling, you would release HF if you use this kind of coupling partner. If you can avoid it, it's again better. So I've not generated the trifluoroboronate salt for this project, and I don't know how it would have behaved in terms of rate in the Suzuki cross coupling. So it's hard to compare in that case. But yeah, concerns around HF liberation was the reason why we didn't investigate that.
It's very fair, thanks.
Okay, probably got time for a couple more questions. So we got a question from Matthew Fawkes. I guess this is a kind of general process chemistry question. Did we buy in the LDA or did we make this? And how difficult is it to handle organolithiums on scale?
Maybe Matt will like to chip in at some point as well on this one because it's quite general. I think so. It would depend on the preference from the contract manufacturing organization that we would pick. I suppose we would have discussions about this. Those are quite typical reagents. So on scale, it's hard to stay away from them for every project. So they are definitely usable on scale. Maybe Matt, you know more about this?
Yeah, so we use lithium HMDS in our process, and we actually used it in TBME as a solvent, which isn't one you'd normally come across. And we found if you want it, there's a supplier out there who will sell it to you. You know, if you ask for 3,000 litres of lithium HMDS in TBME, people will prepare them for you. Suppliers are very good with that and very accommodating. And then when you actually go to our CMOs, they are really good at doing a lot of very anhydrous air sensitive chemistry. In fact, the inertia you get on a pilot plant is probably better than you can get in a lab. So there's not much problems using organolithiums really, or Grignards.
So I think my understanding has always been that it's fine to use it on plant as long as you've got the appropriate kit. It's just if you want to actually develop the process, it's the development which is the problem. That's just what I've been told.
Yeah, I mean, I'm sure if you want to use neat diethyl zinc, someone's going to have a problem with you. Yeah.
So I think maybe in the interest of time, we'll take one more question and then I think we'll wrap up.
I agree with that. So Matt, if you have time for just one more. I'll take a question from Mark Reed at Bristol. So he is asking, with regards to the nitration and reduction strategies, whether instead you explored direct electrophilic amination. So, for example, with benzoxyhydroxylamines, other than the radical mediated example, which I think you did have in there.
Yeah. So we looked at some of the radical stuff, and that's sort of based on the this similar, more modern work by Bill Morandi. And some of his reagents we looked at as well. If you're talking about the electrophilic, so what is it? Is this sort of like the reagents that Bookworld uses for his copper catalyzed amination chemistry? No, we didn't. I think that pyrazole compound we're looking at is relatively electron deficient. I don't think it'd be nucleophilic enough to do a sort of a SNAR type reaction. Yeah,
sounds reasonable.
I think as well, a lot of times when you get into more complex reagents, you've got to factor in the fact that you might have to synthesize them. So that HOSA reagent's great because it's commercially available. When you start making specific reagents, there are additional steps in your synthesis.
Yeah, very much agreed. Might not want to have to do extra steps on huge scale. Cool. Well, I think we better make that our last question here. But those were two absolutely fantastic talks. So thank you guys very much, and thank you everyone for attending. Pass over to Ali.
Yep, so thanks both Matt and Jean-Marc. It's been really, really good talk. It's pretty nice having some colleagues on the line. So next week we've got Lars Barfaker from Bayer, and then the week after we've got Kevin Lamb from the University of Greenwich. So I think just take the last opportunity to thank Matt and Jean-Marc, and we'll see you hopefully next week. Thank you, everyone.
Thanks.
Thanks.